Okay. Hello, everyone. My name is Meng Li, and I am a postdoc working with Dr. Rory Sawyers at Penn State University. So my research interest has been focusing on understanding the below-ground interactions, including root-soil interactions, or rhizosphere microbiome and mycorrhizal fungi to help us improve crop productivity as well as the system sustainability. So my previous research has been focusing on the cropping system level. So now I'm learning more crop genomics tools and then have this genomic part in my story. Today I'm going to talk about root diversity and adaptation. And I would really love to hear any of your questions and suggestions. Okay, so my topic will be detecting the genetic loci underlying root adaptation to environments in maize. So this is actually a collaborative work between Chloe, a PhD student in our lab, and me. Chloe has been doing mostly the phenotypic part of the analysis and some modeling, and she has already presented her part of the work at the evolution about 2 months ago, and you should be able to check her talk through the YouTube channel. I'm mostly focused on the genotypic part of the work, and that's what I'm going to show you today. So basically, we are interested in roots, especially root anatomical traits, because they are very important for soil resource capture and also for plant stress tolerance. So on the left here, you see two pictures showing the maize root anatomy. And these are root cross-sectional images taken by laser application technique. So these two genotypes, they are showing very different root anatomical traits. So some root anatomical traits that we are interested in is, for example, first one is the aerenchyma area. These aerenchyma, they are air-filled spaces that formed inside the root or in the cortical area, and they usually develop under stress, for example, drought, flood, or suboptimal nutrient conditions. And previous studies have shown that the formation of aerenchyma area can reduce the root metabolic costs and allow the root to have a better growth or more growth towards the soil, deeper soil layers, or to explore a larger soil area, and that will help the plant to absorb more nutrients. And similarly, the cortical cell numbers and also the size of the cortical cells are also important to that will influence the root respiration and nutrient content, and also related to the root growth and how they react in terms of stress. Another trait is about the metaxylem. So we are interested in the size and the number of these metaxylem vessels because they can influence the water uptake and water transport in roots. And then previous studies have found that actually reduce the number of metaxylem vessels can help the plant to conserve soil water through the whole growing season, which is called water banking strategies. And using such strategy, the plant can combat, for example, drought. So you can see these traits, they play very important functions that can help a crop performance under suboptimal water and nutrient conditions. And also they are heritable. So previous studies have reported that the broad sense heritability of these traits can range from 0.4 to 0.8. So given that they are important, they have important functions, and also they are relatively have high heritability. Actually, these traits can merit more considerations as breeding targets for improved crop performance under especially suboptimal environmental conditions. Also previous studies have found that there are large phenotypic variation in this landrace root anatomy. So on the left, you can see this picture here, depending on the origin of the landrace, their root anatomy are showing very different structures there. So these landraces, they are domesticated, and they have developed at their local environment for a very long time, and they are considered to be adapted to the local environment. So such high root diversity in this maize land races kind of provide us a great opportunity to understand the genetic architecture that behind this these adaptive root anatomical traits, and also how they interact with the environment. So actually using maize land races has been proved to can help us understand crop adaptation to diverse environments, and also to identify some adaptive genes. And these approaches, like related to identify adaptive genes, can be several, like multiple ways to do that. One way of doing that is to look at the environment genome associations. Basically, it's looking at the correlation between the environmental gradient and the allele frequencies across the genome, and then extract the top or most significant associated genetic loci that can be considered kind of adaptive genes or genetic loci. And another method is to use common garden experiments, basically, or transplant experiments, to grow many land races in a common garden across different environments, and then phenotype their traits, and then we can do the GWAS to extract the top SNPs or genes that are associated with the target of interest, the trait of the interest. So by doing such approaches that have already been proved to very useful and can identify some, for example, flowering related genes like that have been reported to be related to flowering time in previous studies. And also it is proposed that looking at this overlapping genes set, we may be able to get some genes that are both adaptive and related to the trait of the interest. And to look at such genes may help us get a better idea of this adaptive genes, and also reduce the possibility of detecting some deleterious mutations. So such kind of overlapping genes may merit further investigation. So because of, like, given these techniques, our thought was to look at the roots, and was thinking if we can find the environmental associated genes using the landrace environment information, and also have the root phenotypes from these land races, that will help us to identify this adaptive root genes that related to this root anatomical traits, and that would be ideal. But the problem is actually large-scale root phenotyping is very challenging. So you can see some pictures here showing the examples of how root phenotyping can be done. We can do lab or greenhouse phenotyping methods that can give us a relatively high throughput manner kind of result, but these young seedlings and artificial soils that was used in this laboratory or greenhouse experiments may not represent the mature roots in real heterogeneous field conditions. But for the field phenotyping, it's actually very labor intensive. It involves a lot of like digging and washing, and then we get the roots to do further process. And that large scale root phenotyping for the land races are it's actually very limited. So given that the difficulties of like updating large scale landrace root phenotyping results, we were thinking that, is it possible to use some existing landrace and root data resources to help us answer the question? Although these data sets may not be perfect, but they may give us a clue or guide us to a more detailed further of genotyping and phenotyping efforts. So when we are trying to search for available data resources, we found that actually Dr. Lynch's lab has already extensively phenotyped inbred panel for their root traits. And we were able to recover 175 inbred genotypes with their genotypic information, and also 8 root anatomical traits for these inbred genotypes. These traits are like some related to what I introduced in the previous slides. And on the other hand, the land races diversity panel is also there. Like we were able to recover around 1,800 landrace genotypes with their genotypic information, and also their geographic information. So using their geographic information, we were able to extract a lot of climate and soil variables from public data sources, for example, the weather conditions and some modeling work, and to get the environmental variables for each landrace genotype. And this step was done by Chloe. So with that, I just want to kind of recap of what kind of data resource that we have right now is that we have the inbred panel with phenotypic information and genotypic information, but we don't have the environment information for that. For the land races, we do have the genotypic information and the environments, but we don't have the direct phenotypic data for that. And the question is how we can get use of this data. Like we saw that if we could do, for example, a GWAS on the inbred panel, and that may allow us to extract some genetic loci underlying root phenotypic variation. And then if we do a environment, a genome association study, we will be identify some genetic genes or genetic loci that are involved in adaptation. And when we look at the overlapping gene, that may give us an idea of this adaptive root genes. So because these two panels, they were genotyped using different platforms, so we actually compared them based at the gene level. So you can see here, actually most of the genes are shared by these two panels. So our naive thought was to maybe we can just directly compare the results and see what will happen. So here are what we did. Basically for the inbred panel, we did our GWAS using the grid LLM pipeline that actually allow us to extract the genotype May effect and the genotype by environment interaction effect. We use Magma to aggregate the SNP level p-values to the gene level p-values. And then based on the p-values, we picked the top 100 genes for each anatomical trait and pull them together to get a gene list for the inbred panel. So these will represent the root genes from this inbred panel. And for the land races, we used a similar pipeline. This time we used Magma to aggregate them into gene level results, and then also pick the top 100 genes for each environment variable, and then put them to get the top gene list for the environments. So what we get is when we overlap these, we get a 54 genes in this overlapping set. But the question is, how should we evaluate our results? And are these overlapping genes showing some signal of enriched root-associated genes than expected. So in order to evaluate the results, we further compiled a gene list from different published resources that they are primarily expressed in roots, or they have been identified in previous studies, or that is, they are associated with roots. So we included, for example, this genes from this database, and we also included genes that are associated with root development by searching through the maize JDB GWAS track, and extracted around 1,500 genes. And we also included 2 studies that specifically looked at the anatomical traits for roots, and to get more genes in this list. So by looking at this, we compared our results with that compiled root associated genes, and our expectation was that the overlapping genes between these two may contain a higher ratio of the root associated genes than expected. So the expected ratio is here. The dashed line is calculated based on how many root associated genes are in this top gene list pool. And we hope to see like a higher bar here, but in reality, this bar, the ratio is actually lower than the expected. It's not even higher, and it's actually on the contrary. So so several things may explain such kind of results. The first one is, it is possible that most of the genes, if we choose them based on the p-values, they may not be the real genes that are like working. This could be the indirect effect on, they could have indirect effect on the root phenotypic variation, or the interactions with other genes. Another thing is that the signals of local adaptation that have accumulated over hundreds of generations in the land races may relatively hard to capture using our small scale or maybe lower diversity panel, or short-term field trials. So since the direct comparison was not as expected, we used another study in which many land races were phenotyped with the anatomical traits. So in this paper, we were able to recover 39 land races with their anatomical traits, as well as their environment information. But unfortunately, these land races were not genotyped, so we don't have the genotypic information here. So the question here now is how we could maybe combine these existing data resources and help us to do to understand the question that we asked. So here actually, Chloe did a key step to use the environment information of land races to predict their root phenotypes. I will just briefly introduce what Chloe did. If you are interested in more details, you can check her talk on our Z Evolution YouTube channel. Basically, what Chloe did was we first used the published from the Burton data, we have observed root traits as well as their environmental information. So Chloe used a feature selection method to select the most important environment variables that can contribute to the root traits, and then use these environmental variables. She applied a random forest modeling pipeline, and to kind of project project the root anatomy to our 1,800 land races. So here is the example of the results. You can see that our observed values and the predicted values, they align pretty well. So with this step done, actually we can use the root anatomy here and the environmental information and also environmental information of these land races to get a predicted root anatomy for our this 1800 land races. And we now can use this information in our following data set. Also, we applied the same pipeline as we did for the environment to this predicted root phenotype and to get their top gene list. When we compare these two, so this basically this root phenotypes, because they are predicted by environmental variables, so you can consider that as a nonlinear combination of different environmental variables. So when we compare the variables that were used to predict these phenotypes, we found that using these predictions, we were actually able to capture some top genes that were found by using the environmental variables itself, and also a different subset of peaks. So because these predictions are also related to root and also the environment, so that maybe give us a better idea to identify the adaptive root genes. So here I'm just giving a density plot showing the top genes that we recovered from using just the environment and using the predictions. For example, you can see here on chromosome 8, both of our methods are recovering same peak here. So, but on, for example, chromosome 3, we found more top genes in our predicted, using our predictions, but that may be not the case for the environment. So now we have like different values or environmentally based root predictions. And so we were thinking of what if we use this data and we overlap that with the INBRAT panel? Does that improve our confidence to obtain more root-associated genes? So here's the result. So, but basically by overlapping these two gene sets, we find 20 genes in this overlapping set, and actually it is substantially the ratio of root-associated genes in this overlapping list is substantially increased, comparing if we just directly overlap the environment and the root anatomy from the inbred panel. Although the p-value is not significant here, it could be partly due to the small sample size here, because we only have 20 genes in this overlapping gene set. So that could be some randomness or random errors are playing the role there. So in order to rule out the potential randomness, we also kind of applying the same method to using different sample top genes. So far we've been just using the top 100 genes, and now we use the genes from the top 20 genes until top to 500 genes, and then get the expected ratio and also the ratio of root-associated genes in our low overlapping gene set. And you can see in the overlapping gene set, we always got a like higher ratio of root-associated genes than expected. So that means by using this environmental-based root predictions, we may be able to get, that may help us get some good results that are both related to root and also adaptive to the environment. So our following studies are mostly focused on this using these predictions in the GWAS. And previous studies have discussed that within this 200 kb region, you may like more possible to find the real gene that is in action. So instead of like looking at the gene, individual gene level, we then add 200 kb to our top genes from this using the predictions, and then um, to look at the gene regions. And if two regions, they have overlaps, we then merge these regions together. So by doing this step, we actually end up having 445 regions. We further filtered these regions based on two strategies. The first one is we want at least one genes that were the top genes from the inbred root GWAS, and also or at least one genes were found in previous published studies that are related to root-associated genes. So by doing that, we further filter these regions down to 210 genes. We actually found many interesting genes by doing this step. And I will give 3 examples in the following slides. So the first thing I want to mention that using the pipeline I just mentioned, we were able to recover many genes that have been reported in previous studies that they are involved in root growth or root development. For example, this acetylene response factor that they have been characterized in previous studies that are very important for root development and the formation of lateral roots. And also other genes has been reported in previous studies. Another point I want to point out that using like both the landrace and the inbred GWAS results, that may help us to kind of have a better resolution to identify the adaptive genes. So here on the left, you can see it's a Miami plot showing this region the p-values of the genes. So on the top, these are for the land races, and on the bottom, these are for the inbred lice. And on the top panel, the yellow points are for the environment genes, and the green ones are for the predicted root genes. And those red circles are actually the genes that are in our top gene list. So here you can see actually we found, for example, here, these genes, they are very closely next to each other. And when we look at this gene, for example, here, we find that it is highly expressed in roots, especially in the steel area. It is actually following our predictions. We also found that the top gene for the metaxylem traits. And it is also proved to be very important in Arabidopsis, that they are associated with root development. And another 2 genes here, next to that gene, they were also reported to be important for metaxylem area underwater stress, or they being captured in that root-associated gene databases. So that means like in this area, there may be possible or real gene, or at least one adaptive root gene is there. So we then looked at the LD heat map at this focused area and compare the inbred with the land races. So using the inbred LD heat map, you can see they are showing a relatively high LD. That means just using the inbred panel, we may not be able to tell apart like which gene maybe like the candidate gene, like, because they are all showing similar p-values. By using the landrace panel, it's hard to see here, but they have very low LD, and meaning that we may be able to tell like maybe these 2 genes are not the same thing. They are both important, something like that. So another point, like I want to discuss is that using our environmental based predictions to do the GWAS, we were also able to identify some important environmental related genes that cannot be captured by just looking at the inbred panel results. Here you can see this red circles here, the top genes that we found in our environmental gene list, and also using the predictions here. And this gene was, when we search the maize JDB, it is highly expressed in roots, and it was reported in a previous GWAS study that it is related to root development. And in Arabidopsis, it is also an important gene related to root development. And when we look at one SNP, for example, the most significant SNP in this gene, you can see a clear latitude distribution of the minor and major alleles here. But for using our inbred panel, we will not be able to recover that gene. So that means using our predictions, we may also able to recover some important environmental adaptive genes that is also root candidate genes for important root involved in root development. So that's basically, I want to summarize what we did. We first used different data sources using try to understand the genetic architecture of root anatomical variation using land races, inbred lines, and also the land race environments. And we, Chloe developed generated a pipeline to use the environment to predict the root anatomy. And using that predictions, that actually gave us a greater ability to adapt it, to identify these adaptive genes. Or if we combine that with the inbred panel GWAS results, that can help us to identify some potential important root genes that can also maybe adaptive to the environment. And there are also some ongoing future steps. By collaboration with Carlos from Mexico, we will have the single cell expression data. So that can kind of give us a more detailed validation of our anatomical traits. And also we used we are going to use a magical population to test the hypothesis of trait environment adaptation and to identify the genes that we identified here and see if that will also show up in the mapping population. So this work is in collaboration with Peng from Germany, and we actually applied the same pipeline to seminal root numbers and identified some very interesting candidate genes there using this approach. So we will be applying this to our other anatomical traits that I just mentioned. So with that, I would really like to thank all my lab members, previous lab members and current lab members, and also our collaborators. And thank you for your attention. I would love to hear all your suggestions. Thank you.
Okay, thank you so much, Meng. I have written myself a couple of questions in the chat, so Rui sort of answered, but anyway, maybe you can add some details. I don't know. I was wondering whether when you use the environment to predict the phenotype, do you assume that there is no G by E interactions? This was not so clear to me. So Rui said no, but
you mean using the environmental predictions?
Yeah.
No, actually, like the G by E was not like in that pipeline, but I think we can like Chloe was maybe able to predict some G by E effects if we have some common garden like results from root phenotype that have root phenotype data that we may be able to use. But so far, like using current data, we don't have G/E effect.
Yeah. No, I am not asking that. I was asking whether by relying on the environment to predict a phenotype, I had the impression that you consider that there is no G/E, but
I think what we're predicting, Maud, is how things from those different environments would perform in our common garden, or for instance, in our greenhouse. So although we display it on the map, which is very nice, we're not actually saying that that's what you would see if you went there. We are saying if you collected corn from those places, and for instance, the training set we used was greenhouse data. If you brought those varieties to the greenhouse, that's what you would see. So certainly, yes, to then extrapolate back out to the environments, we would need to do something a bit different. So again, we're
no, I understand that, but then this has sort of assumed that you don't have G/E, no? Because if you had G/E, you could really not predict this genotype behavior in another environment.
So that
you have to assume that there is no G/E.
That would mean if we collected a load of land races and did them in common garden, say in the greenhouse, they would all look the same. If everything was purely driven by G/E, which could be the case. So I think we're not saying we're necessarily capturing everything, but I think we have enough data to suggest there is enough main effect that we can see variation clearly in common garden. And it does seem to map back to meaningful environmental gradients.
Okay. And then
it's probably more extreme in reality. No?
And then the other thing is the predictions you showed, I mean, they were correlated somehow with so you had a prediction, I think it was 34% of the variation that it explained, right? But this is not, I mean, this is not super accurate, right? I mean, it's not like
I think that's true. I mean, obviously the training sets are small.
But then I was wondering when you
increase them, it will get better, no?
Okay, can you go back to your different sets? I think there was a slide because.
Sure. Am I sharing my screen?
Yes, for instance, the slide 17 or something. I don't know.
This one?
Or maybe.
Or this one?
Yeah. So, for instance, no, the one before. The one you just
17.
showed.
The one down, Meng, the one with the next table.
16?
17.
Okay.
17. Okay.
Yeah. Okay, so here you were predicting the foam diversity panel, right? And okay, because in the Burton panel you found a prediction power. I mean, so you captured like 34% of the variation or something like this, right?
Of that trait.
Yeah.
For that trait. But in the foam diversity panel, I mean, these two panels may not be. Okay. No. Maybe.
I mean, the thing about the Burton panel, it is Mexican land races. So, and it's some of the material, it doesn't quite overlap, but it is comparable. Obviously, the inbred panel is something completely different.
Okay. I understand. Okay.
Something we're doing, which is, again, it's not trivial work, is we're at least regrowing some land races to see if they follow our predictions. So Chloe's doing that now.
Okay.
But again, for us to do 8-10 land races with replicates, you have to take different routes, section different routes. It's all, it's quite demanding. So I think at this point, what we're suggesting is that there is a pipeline here which seems to have some explanatory power. And obviously, if we add more training data, if we increase the sampling that we can put in, obviously that will also go. And we're not saying this is a one-off. I think it's a cyclic process of informing ourselves, and the more data we add, the more power we will have. I think the concept, though, is that unlike flowering time, we're never going to go and measure 1800 land races for roots.
No, absolutely.
We need other ways to allow us to explore this diversity, which is accessible. And I think these modeling, which could be more or less sophisticated, is a way to get there.
Yeah.
Okay, thank you.
Maybe add to Rory's point, because we used the same pipeline for seminal root number in collaboration with Peng from Germany. And actually, he phenotyped a lot of the land races with the seminal root number. And using this pipeline, we actually recovered some very interesting signals there.
Yeah. Okay, thank you. Are there any comments or questions of the people around? I didn't see other questions in the chat, but please.
I have many questions.
Yeah, go ahead.
Sure.
So I see you used multiple environmental variables. So the first question is, you did like a joint analysis of multiple phenotypes, or you did multiple different genotypes independently for those genome environment analysis?
I just want to make sure I understand your question, that you asked that I did kind of hold. So for example, for these, let me see, how can I find this? Yeah, so here, but for example, we have 8 root anatomical traits for the inbred panel, and we have like 39 environmental variables for the land races. And the top gene list was compiled by extracting the top 100 genes from like all of 8 anatomical traits. And for the environmental gene list, we extracted the top 100 genes from all these 39 variables and then put them together.
Okay, so every climate variable and soil variable, every environmental variable was an independent model. You run everything independently.
Yeah,
Yes.
so yeah, we run everything separately, but they do have a lot of overlaps. So like if everything is separate, we will be able to get like 3,900 genes, but in the end, we only found like, for example, 1,300 genes. So a lot of this environmental variables, they share some same gene signals.
And the second question now, what was like the environmental variables that had the most genes on your list?
Wow. I think precipitation and temperature related variables showed the most like significant kind of genes, and soil variables we actually didn't like, maybe not as that important as the climate related variables in our data set.
Thank you.
Yeah.
Okay. Do we have other questions? Oh, Ed.
Yeah, I was kind of wondering how much or how stable are the soil related variables then? I mean, has there been enough change in the last, century of agronomic practices, by farmers, that what we're measuring as the soil variables don't match very well with what the genetics is that's coming from? Is that possible, or?
Yeah, that's possible. That's a good question. I think the variables, soil variables that we used are from this published like modeling work, and we also kind of averaged from, I forgot it's from 1920s until like 2000. So it's like an average of many years of data. But it is possible that the soil can really change due to the like agricultural practices and other like climate change. But I think the idea is that like using the land races, we are capturing the big gradient of the soil variables that kind of can help us understand some of these. We are not like really going into a lot of details into the soil variables here.
Yeah.
Okay, thank you, Ruben.
Yeah, following up on that, have you tried doing anything with teosynthesis?
No, not with teosynthesis. Like,
because there you would expect less changes in soil variables.
Yes.
I think that with the land races, you probably haven't seen a lot of changes in terms of the amount of fertilizer, etc., that is added. I don't know what others think on the audience.
Okay, yeah, that's a good point. Maybe we can look into data sets that, like, if teosynths have such kind of information that we can use. I think like Peng has some information about teosynth on the seminal root numbers, and we may be able to use that to try to understand the question.
I think also we get quite a lot of confounding variables, clearly. I mean, we're working within Mexico, so the highlands are where we find low phosphorus, no? Where you have high precipitation in the south, it's wet, it's warmer. You also have obviously less nitrogen as things wash away, etc. So possibly, again, we're not necessarily capturing soil signals because they are confounded in other possibly stronger signals which we relate to climate, which is, again, something that would potentially we break down when we start moving from the per se land races back into the mapping populations, and again, you know, evaluations under controlled conditions, and trying to break some of this all up.
Okay. I was reading the chat. Okay, so is there any other question or comments? Okay. Okay, so if not, I think we can end up here. And thank you very much again for this interesting talk. Bravo!
Yeah, thank you all for the questions and suggestions.
Thank you, Meng.
Okay,
Thank you.
See you